Okay, let's jump right into a topic that's, well, one of the toughest challenges in oncology. Extensive stage small cell lung cancer, or ES-SCLC.
Right.
This thing is incredibly aggressive, grows fast, spreads quickly, and honestly, the numbers are stark.
They really are. That 5-year survival rate, it's stuck below 7%.
Yeah.
It just screams out that there's a huge unmet medical need here.
Absolutely. And for ages, the go-to first treatment has been chemo. Etoposide plus a platinum agent, EP.
The EP regimen, standard stuff.
But even with that, median overall survival, how long patients lived on average, was stuck around just 10 months. It just wasn't enough progress.
No, not really. And look, there have been some recent steps forward. Adding immunotherapy, specifically PD-L1 inhibitors to chemo, has helped.
Right, nudged survival up by maybe, what, 2 months or so?
Roughly, yeah, compared to chemo alone. But it's interesting because other attempts using PD-1 inhibitors, specifically in this first-line setting, they've had, let's say, mixed results before, not always consistent wins.
Which is the perfect lead-in to our source material for this deep dive. We're going to unpack a big, recent phase 3 trial. It's called EXTENTORCH, published in JAMA Oncology, a top journal, and it looked at adding toripalimab, that's a newer PD-1 inhibitor, to the standard EP chemo for folks with newly diagnosed ES-SCLC.
Exactly. So our mission today, really, is to walk you through what this trial found. We'll look at the design, the key results, survival, progression, the safety side of things, of course.
There's some really fascinating biomarker stuff, too, right?
Yeah, definitely. We'll get into that. The goal is for you to come away with a really clear picture of what this toripalimab combo offers and, what it might mean practically.
Okay, let's start with the basics. How is the trial actually set up?
So this was a pretty large study, multi-center, double-blind, placebo-controlled, the gold standard, really. Phase 3.
Right.
They took patients with newly diagnosed ES-SCLC who hadn't had treatment before and randomly assigned them. Half got toripalimab plus the EP chemo.
And the other half?
The other half got a placebo, a dummy drug, plus the same EP chemo.
So everyone got the standard chemo backbone, but the immunotherapy was the key difference.
Precisely. They went through up to 4 to 6 cycles of this initial combination therapy. Then came maintenance.
Maintenance.
Yeah, the toripalimab group kept getting just toripalimab, and the placebo group kept getting just the placebo. This continued until, well, the disease got worse or side effects became too much, or they hit the 2-year mark.
Makes sense. And what were the main things they were measuring, the primary endpoints?
2 main things. First, progression-free survival, or PFS, basically, how long patients lived without their cancer growing or spreading, as assessed by the investigators.
Okay.
And second, the really crucial one, overall survival, OS, just how long patients lived, period. They also did a lot of digging into tumor characteristics, biomarkers, trying to see who benefited most.
All right, let's get to the results then, the headlines. Starting with PFS, did adding toripalimab help keep the cancer in check for longer?
Yes. Unequivocally, yes. The trial hit that endpoint. Adding toripalimab significantly improved investigator-assessed PFS.
How significant are we talking?
Well, the risk of the disease progressing or death was cut by about a third. The hazard ratio was 0.67.
Okay, 0.67.
And the p-value was tiny, less than 0.0001. So very statistically significant, highly unlikely this result was just chance. Remember, a hazard ratio under 1 means a lower risk.
Right. So what did that translate to in terms of actual time, like median months?
So the median PFS, investigator assessed, was 5.8 months for the toripalimab group and 5.6 months for the placebo group.
Huh. Only 0.2 months difference in the median. That seems small.
It does look small if you only focus on the median. But, and this is really key, you have to look at how the survival curves behaved over time. They separated.
Ah, okay. So more people stayed progression-free later on.
Exactly. Look at the rates. At 6 months, it was 47.1% progression-free in the toripalimab arm versus 36.3% in the placebo arm. But look at 1 year, 18.1% versus just 4.9%.
Wow, that's a big difference at 1 year.
It is. That later separation really shows that a subset of patients seems to get a more durable benefit from the immunotherapy combination, even if the initial median isn't dramatically different.
Okay, that makes more sense. So it's helping some people stay well for much longer. Was this PFS benefit seen across the board, like regardless of other factors?
Yes, and that's important. The benefit was consistent. It didn't seem to matter if the tumor expressed PD-L1 or not, and it held up across the other main patient subgroups they looked at.
That's good to hear. Okay, now for the ultimate measure, overall survival, did people actually live longer?
Yes, and this is where it gets, I think, very clinically meaningful. Adding toripalimab significantly cut the risk of death by 20%, actually.
20% reduction in risk of death.
That's right. The OS hazard ratio was 0.80, and again, statistically significant with a p-value of 0.03.
And the median survival times?
Median OS was 14.6 months with toripalimab versus 13.3 months with placebo.
Okay, so an improvement there too. And like with PFS, did the curves keep separating over time?
They did. You saw it at the 1-year mark. OS rates were 63.1% versus 54.9%, and even more so at 2 years. 25.9% alive in the toripalimab group compared to 19.5% in the placebo group, consistently favoring the combination.
So clear wins for both PFS and OS compared to just chemo. But hang on, that median OS in the placebo group, 13.3 months. You mentioned earlier the historical benchmark was around 10 months. Why might it be higher here?
That's a really sharp observation. The authors noted that too. One likely factor is what happened after patients progressed on the initial trial therapy.
Subsequent treatments.
Exactly. A larger chunk of patients in the placebo group went on to get other treatments later on, including potentially other immunotherapies like PD-1 or PD-L1 inhibitors, or drugs like Anloctim, which is approved in China for later-line SCLC.
Ah, okay, so that might have boosted the survival in the control group a bit compared to historical data?
It could have, but the researchers trying to account for this. They did what are called sensitivity analyses, adjusting for these subsequent therapies. And even after those adjustments, the upfront toripalimab combination still showed a significant survival advantage.
Got it. So the initial benefit seems real, even with that potential crossover effect. What about shrinking the tumors, response rates?
Yeah, objective response rate confirmed tumor shrinkage was numerically a bit higher with toripalimab, 78% versus 73.1%. But that difference wasn't statistically significant. Disease control rates were pretty similar too.
But maybe the responses lasted longer.
That's the key. The duration of response was indeed better in the toripalimab group. That fits perfectly with the PFS curve separating later on.
Makes sense. Okay, let's shift gears to safety. Adding immunotherapy usually means different side effects. How did this combo stack up?
Right. Always a critical question. Interestingly, if you look at the overall rate of any treatment emergent adverse events, or even the severe ones, grade 3 or higher, the percentages were actually very similar between the two groups.
Really? Similar rates of severe side effects?
Yeah, around 89–90% had some kind of side effect in both arms, and about 64% in both arms had a grade 3 or higher event. Surprisingly close.
So where did the differences lie?
The main differences popped up with serious adverse events. Those were higher with toripalimab, 50% versus 37.5%. And, as you expect, with immune-related adverse events, the IRAEs.
The specific side effects caused by revving up the immune system.
Precisely. The investigators flagged IRAEs in about 29% of patients getting toripalimab, compared to only about 11% in the placebo group.
What kinds of IREs were most common?
Mostly things like thyroid issues, hypothyroidism, hyperthyroidism, and also pneumonitis, which is lung inflammation. Those are fairly typical for PD-1 inhibitors.
And severe immune side effects.
Grade 3 or higher IREs were also more frequent with toripalimab, about 10% versus less than 1% with placebo. So yes, more immune issues, but the trial investigators felt the overall safety profile was manageable and in line with what's expected for chemo plus PD-1 blockade.
Okay. Now, this biomarker analysis sounds really intriguing. Trying to figure out who benefits most. What did they find digging into those tumor samples?
Yeah, this is pretty cutting-edge stuff. They did whole exome sequencing, looking at the coding DNA on samples from about 300 patients, a big chunk of the trial population.
So did something like tumor mutational burden, TMB, predict response? We hear about that a lot in immunotherapy.
You do, but surprisingly, not really here.
Yeah.
In this study, they didn't find a significant link between TMB status, high or low, and how well patients did on toripalimab for either PFS or OS.
Huh. Okay, so TMB wasn't the magic bullet predictor in this case. What else did they look at?
They found some interesting signals with specific gene variations or mutations. For example, genes like CAMT2D and COL4A4.
How did they matter?
It seemed the patients who had the standard or wild-type version of these particular genes actually did better on the toripalimab combo compared to patients who had variations in them.
Okay, so for those genes, having the normal version was better for immunotherapy response.
Appeared to be, yes, in terms of PFS and OS benefit from adding toripalimab.
Were there other genes where having a variation was actually linked to a better outcome with toripalimab?
Yes. Interestingly, the flip side seemed true for genes like CTNNA2 and SCN4A. Having specific variations in those genes correlated with improved PFS and OS, specifically for patients getting the toripalimab combination.
Wow, okay, so it's complex. Some gene changes might hinder the response, Others might enhance it. It's not a simple story.
Not at all. It really highlights the intricate biology involved.
Were there any broader tumor features beyond single genes that seemed important?
Yes, two really stood out in the exploratory analysis. One was about tumor heterogeneity.
Meaning how different the cancer cells are from each other within the same tumor.
Exactly. And what they found was that patients whose tumors had low intratumor heterogeneity, meaning the cancer cells were more genetically similar to each other, those patients had significantly better PFS and OS when they got the Toripalimab combination.
Low heterogeneity correlating with better immunotherapy response.
That's fascinating. Maybe a more uniform target for the immune system.
That's the leading hypothesis. If the tumor cells are all singing from the same song sheet, maybe it's harder for them to hide or evolve resistance when the immune system gets activated by the PD-1 inhibitor.
Okay, that makes intuitive sense. And what was the other major finding?
It involved HLA haplotypes. HLA genes are crucial. They control how cells present antigens, those little flags that tell the immune system, attack here.
Right, the immune system's presentation machinery.
Essentially, yes. And they found that patients carrying a specific HLA combination, HLA element positive and HLA B62 negative, seemed to get particularly prolonged benefits from the Toripalimab treatment.
So your specific immune system type, based on HLA genes, might influence how well you respond to this therapy. It certainly suggests that. How effectively the tumor's wanted posters are shown to the immune system, or maybe how that person's immune system is naturally tuned, could be influenced by these HLA types. They did note that HLA-11 is more common in East Asian populations, which is where this trial was primarily conducted.
Okay, so putting the biomarker pieces together, it looks like, although it's early days, tumors that are less varied internally maybe have certain specific gene profiles or particular HLA types for antigen presentation. These might be the ones getting the most durable bang for the buck from adding toripalimab.
That's definitely what this exploratory data points towards. It needs confirmation in other studies, for sure. But it's exciting because it hints at ways we might, down the line, be better able to select patients most likely to get that really significant long-term benefit.
Right. So let's try and summarize the main message from this deep dive on EXTENTORCH for everyone listening.
I think the core takeaway is pretty straightforward. This phase 3 trial clearly showed that adding toripalimab, this novel PD-1 inhibitor, to standard first-line chemotherapy significantly boosted both progression-free survival and, importantly, overall survival for patients with extensive-stage small cell lung cancer.
A clear improvement over chemo alone, and on the safety side.
Yes, there were more immune-related side effects, which you expect with this class of drugs. But the overall rate of severe side effects wasn't drastically different between the arms, and the safety profile was deemed acceptable in the context of the potential benefit.
So this deep dive has really walked you through the evidence from a major study that supports this combination as a potential new standard of care, a new first-line option in a disease that desperately needs better treatments. Okay, here's a final thought for you to chew on. Building on what we've discussed, particularly those biomarker findings, we saw hints that things like low tumor heterogeneity, specific gene variants, even HLA types might predict who gets the biggest benefit from toripalimab. So the question is, what could understanding these really complex genetic and cellular differences actually mean for the future of treating ES-SCLC? How might unraveling this biology lead us towards even more personalized, more precisely targeted, and ultimately more effective therapies for these patients down the road?